أهلا وسهلا بكم في مؤتمرنا التاسع لاسم الأشعة. أحب أشكر جدا اللجنة المنظمة، الحقيقة بيشتغلوا جامد جدا جدا من نهاية المؤتمر السنة اللي فاتت علشان يطلعوا المؤتمر بصورة مشرفة إداريا وعلميا وتنسيقيا وكل حاجة، وعلى رأسهم أستاذنا الكبير دكتور محمد أبو الهدى. بجد بشكرهم شكر جامد جدا جدا. أحب أرحب طبعا بكل الحضور وبالسادة المشرفين النهاردة as chairpersons in this first neuroradiology session. الدكتور الجنرال الكبير الدكتور هاني حافظ، ودكتور برضو محمد عمرو في الأكاديمية العسكرية، الأستاذ الكبير الدكتور خالد عصمت، ودكتورتنا الجميلة الأستاذة الدكتورة إيمان جنيدي. كلهم in their own right very esteemed professors in their subspecialty of neuroradiology. This first session is really very interesting. It's all about the lymphatics, the cerebrospinal fluid, and the interstitial fluid surrounding the brain. It's a very novel subject, and I'm very happy that we have researchers working in this field.
Thank you.
It's actually, certain diseases are really affected by this, and if we can image this properly, we can detect early Alzheimer's disease, for example,
Yeah.
and also other things like the idiopathic normotensive hydrocephalus, and also cerebrovascular ischemic lesions.
Yeah.
So it's really very interesting. I think not long ago that this lymphatic system was discovered in 2012 or something, صح?
2012 in mind.
But this is really a novel subject and a very interesting one. The whole session is all about the circulation around the brain, whether the interstitial fluid or the lymphatic system. And we all thought that there were no lymphatics in the brain, but actually now we found out that there are. So I'm very happy to introduce Dr. Maha El Noubi, our young lecturer in our department, very promising and very active, and with her subject is about ثانية واحدة بقى لأنه طويل جدا. The concepts in intracranial interstitial fluid transport and the lymphatic system, anatomy, physiology, imaging techniques, and clinical applications. تفضلي يا
Good morning. I am Maha El Noubi. Today we will talk about concepts in intracranial interstitial fluid transport and the G lymphatic system. What are the ILOs? First, identify distributions of intracranial fluids. Identify CSF production, circulations, efflux, definition of the G lymphatics and its function, conditions affecting the G lymphatics and pathology, diagnostic imaging of the G lymphatic system. Introduction. Intracranial fluid transport and the G lymphatics plays a crucial role in maintaining brain homeostasis and waste clearance. So, understanding these processes is essential for advancing neurological researches, developing potential treatments for multiple brain disorders. Intracranial fluid distributions. CSF represents about 10% of the fluid. Plasma within the vascular system, 10%. Brain parenchymal volume overall, 80%, which is distributed as 68% intracellular fluid and 12% interstitial or extracellular fluid. Monro-Kelly's rule holds that within the cranial vault, there is a reciprocal balance between these volumes, the volume of blood, brain, and CSF. CSF production. It is produced by the choroid plexus within the ventricular system and the ependymal cells. And there is a net CSF movement through the paired lateral ventricles, through the foramen of Monro into the third ventricle, then aqueduct of Sylvius, fourth ventricle. And there is an additional through the foramen of Magenti into the subarachnoid space, foramen of Magenti and foramen logica. CSF demonstrates oscillatory motions related to the cardiac cycle and respirations. Also, CSF flows around cerebral perforating vessels. What is the cerebrospinal fluid efflux? CSF exits the central nervous system via lymphatics or the venous system. Arachnoid villi is the only known pathway for CSF drainage directly into the bloodstream. Perineural drainage to the peripheral lymphatics as through the olfactory nerves and spinal nerves. Spinal lymphatics drain the dura mater in mitra microcircuit that connects to lymph nodes and thoracic ducts. Newly discovered the meningeal lymphatics, or what is known as the G lymphatic system. The question is, does the brain have a lymphatic system? What is the G lymphatic system? We all know that outside the neuroaxis, there is a peripheral lymphatics, communicates the leaky capillary bed to maintain homeostasis by delivery of nutrients and removing waste products. But inside the neuroaxis, capillaries are surrounded by tight junctions that restrict molecular transport. And that's why we thought for many years that there is no lymphatics for the brain. G lymphatics or glial lymphatics, it is first described in 2012 when a group of researchers at University of Rochester used two-photon microscopy with fluorescent tracers to visualize CSF flow from subarachnoid space through the brain parenchyma in a living mice. This results in the discovery of the G lymphatics, reflecting the key role of the glial cells with the function of analogous conventional lymphatics. In their experiments, CSF was entering the arterial paravascular spaces, combining with interstitial fluids and parenchymal solubles, and at the level of the capillary beds, existing venous paravascular spaces. Recent studies discovered that lymphatics is present also in humans. It is a macroscopic waste clearance system that utilizes a unique system of perivascular channels to promote efficient eliminations of soluble proteins and metabolites. It also distributes non-waste compounds, such as glucose, lipids, amino acids, and neurotransmitters. It functions mainly during sleep, and it is largely disengaged during wakefulness. It's mainly composed of three parts, as we said before. Para-arterial CSF influx, which enters both in subarachnoid space, enters the brain parenchyma through the Virchow's rapin, and surrounded by astroglial cells, with endophase containing aquaporine 4, and the efflux is through interstitial fluid efflux perivenous space. What are the conditions affecting the brain lymphatics? There are multiple conditions affecting brain lymphatics, such as lifestyle, sedentary life, obesity, circadian disturbance, sleep quality, aging, stress, substance abuse, genetics such as aquaporine expressions, localizations, and genetic phenotypes, pathology such as hypertension, vascular diseases, traumatic brain injury. All of these conditions result in decreasing lymphatic output, which results in more accumulations of waste products, which leads to neuroinflammation and neurodegenerative disease, which more increase in the protein and waste products, which leads to more decrease in the lymphatic output. What are the waste products like tau proteins, beta amyloids, and multiple other waste products? So the lymphatic dysfunctions has been demonstrated in multiple neurodevelopmental and in neurodegenerative disease, such as stroke, vascular disease, hydrocephalus, CSF pressure disorders, tumors, epilepsy, demyelinating, and even neuropsychiatric disorders. Diagnostic imaging. Multiple radiological techniques have been applied to the G lymphatic imaging, but MRI is the leading modality, as it utilizes non-ionizing radiations and wide variety of sequences, and which can distribute, investigate various tissue and flow properties. MRI approach neurofluid evaluation can be classified by location, brain or spine, compartment, blood or CSF, use of contrast or not, techniques, anatomic or diffusions. First, assessment of the structure, such as the perivascular spaces. Perivascular space imaging can be performed with the standard T2 weighted images. Several approaches also exist to visualize small cerebral arteries, such as MRA and SWI. Artificial intelligence algorithms have been developed to automatically segment the perivascular space, calculate the total volume, density, and tortuous tube length. Overall, perivascular spaces decrease with normal aging process, with disease process, and loss of blood-brain barrier integrity. Diffusion-weighted images and diffusion tensor images. It measures the molecular diffusion of water using various gradients of p-values, already calculable ALPS along the perivascular space index, which can detect perivascular spaces on the axial images at the level of the body of the lateral ventricles and the corona radiata. On the x-axis, the medullary vessels passes on the x-axis, while the association and projection fibers usually pass on the y-axis and z-axis. So the lymphatic functions can be estimated by the relative flow along the perivascular space compared to the projection and association fibers. Overall, the LPS values are inversely correlated with the normal aging disease. Magnetic resonance elastography. It utilizes external vibrations to estimate tissue stiffness. In hydrocephalus, increasing intracranial pressure correlates with increased brain tissue stiffness. These conditions presumptively result in decreased G lymphatic functions. aging and disease studies have demonstrated correlations between arterial stiffness and greater perivascular space burden. Assessment of the flow can be assessed by non-contrast perfusions or contrast perfusion. Non-contrast such as ASL, arterial spleen lippening. Normal choroid plexus cerebral blood flow is several times in choroid plexus higher than in cortex, indicating water transparent from the arterial blood to the CSF. Choroid plexus and the gray matter perfusions appears to follow an inverse relationship in both healthy and diseased patients, suggesting underlying homeostatic regulations and compensation. Another one, phase contrast imaging, which commonly utilized with 2D or 3D directional velocity encodings to measure rapid flow of velocities of CSF and large vessels. Here is a scout for where we take a flow at the level of sylvian ducts, C2, C3 subarachnoid spaces, and also we can take the flow from the large vessels such as internal carotid arteries and vertebral arteries. POLS are what is known blood oxygen level dependent functional MRI. It assesses differential T2 star signal between oxyhemoglobin and deoxyhemoglobin to detect cerebral blood flow. In healthy neuroglial vascular units, increase the regional brain activity will result to increase the blood flow. So CVR or cerebrovascular reactivity mapping can be informing using a vasoactive challenge, like carbon dioxide, breath holding, intravenous acetylzolamides, which induce vasodilations. Normally, the intact neuroglial vascular units, there is a coupling with increased cerebral blood flow, while in these the neuroglial vascular units, the vessels are unable to dilate and appropriate normal cerebral blood flow response is absent or blunted. Here we can show that after the patients have taken carbon dioxide, there is increased POLS activity or coupling. This is a normal person. Loss of this coupling indicates that the neuroglial unit is affected. Cerebrospinal fluid contrast. Direct injection of contrast into the CSF using a minimally invasive percutaneous approach. Access can obtained by various locations along the neuroaxis. Myelography, if there is intrathecal access, cysternography via cisterns at the level of C2, C3, or ventriculography through the ventricles. After injection, the patients have repositioned with a short time delay to contrast to disperse through the CSF outline. Actually, we use sequential T1 weighted MR myelography. In normal subjects, if we demonstrate gadolinium ascends up in the spine to the basal cistern and over cerebral convexes with delayed parenchymal enhancement. Normal gadolinium of GAD penetrates centripetal from the meningeal surface into the cortex and subsequently deep tissues, supporting the G lymphatic hypothesis. In patients with normal pressure hydrocephalus, there is a marked abnormal CSF flow and there is increased flow surrounding the ventricles, which means that there is altered bulk flow and decreased perivascular flow. And here is an example. In normal patients, and patients with normal pressure hydrocephalus, we can see that there is a periventricular distribution of cerebral blood flow and increase even after 24 hours, which is abnormal flow. Conclusion, the G lymphatic is the analog of the lymphatic system in the CNS. As such, the lymphatic system plays a key role in regulating directional interstitial fluid, waste clearance, potentially brain immunity. Thank you.